we are up to our next speaker, who is Dr. Ibrahim Elsharkawi. And there he is. He's an assistant professor of genetics and pediatrics at the Icahn School of Medicine at Mount Sinai, and he'll be discussing nutrition and mitochondrial disease and new insights in recent years about links between the gut microbiome and the mitochondria. I just want to remind everyone that you can put your questions in the Q&A and that our speakers have been answering those in writing. I know we're moving really rapidly, but they have been kind enough to spend the time writing back to you with those questions. So please feel free to continue doing that, and I will pass it to Dr. Elsharkawi.
So what is the gut microbiome? Essentially, this is all the bacteria, fungi, viruses, all the trillions and trillions of microbes that reside in our gut and actually control a lot or are associated with a great deal of physiological processes in our body. And so gut microbiome diversity is important. So the more variation we have in the microbiome, the better it is in terms of our endocrine health, our nutrition, our gut health, our brain health through the brain-gut axis. So the gut microbiome is involved in many different physiological processes. It's involved in nutrition, digestion, and metabolite regulation as well. A diverse microbiome can also help us produce more neurotransmitters like serotonin and dopamine. And when we have an imbalance or a lack of diversity in the gut microbiome, we call that a dysbiosis. And a dysbiosis has been linked to, outside of mitochondrial disease, many different disease states like inflammatory bowel disease, diabetes, cardiovascular disease, and even mental health issues like depression and anxiety. So just a few terms to go over before we dive into what the research has shown in gut microbiome and mitochondrial disease specifically. So when we talk about alpha diversity, this refers to the variety and abundance of all the different microbial species within a particular sample. So a lower alpha diversity, meaning lower diversity of organisms overall, is associated with dysbiosis, so an imbalance in the gut microbiome. So alpha diversity, when we think about alpha diversity, we're thinking about both the richness, how many different species we have, and evenness, how evenly the species are distributed as well. And another measurement we can take into account when we're thinking about these things is Shannon diversity, which is a measure to quantify the diversity of species in the community. And in the gut microbiome, this evaluates both richness and evenness. So these are two numbers we look at when we're studying the gut microbiome in general. So we've talked about how the gut microbiome, dysbiosis and an imbalance in the gut microbiome has been linked to many disease states. So what about in mitochondrial disease specifically? So this concept started in part by this study in 2019, which looked at the gut microbiome in mice with different mitochondrial genotypes. So I'll walk you through it. So there are they have two types of mice here with common mitochondrial DNA variants. One that is associated with greater ROS production, reactive oxygen species, than the other. What they did here is that they cross-fostered the pups. So as soon as the pups were born from this mom, let's say that the red mice, they went over to nurse and cross and be raised essentially with a foster mom, the purple mice, for the sake of this diagram here. And then vice versa. The purple mice's children, offspring, went immediately to be fostered by the red mice here. What they found was initially the gut microbiota of the pups resembled their foster moms, because that's the mom they were nursing from. That was the close contact that they were experiencing. So initially, gut microbiome resembled mostly that of their foster mom. But interestingly, over time, over the course of 2 months, the gut microbiota of the pups gradually reverted back to that of their biological moms, and they looked different as well. So that's telling us here that the gut microbiota are actually greatly influenced by the host mitochondria, by the underlying mitochondrial DNA, and that ROS may play a part here. And they took this a step further, and what they did was that they decided to manipulate the degree of reactive oxygen species production. So they did this by aging the mice, which increased the reactive oxygen species, treating with NAC, which, as many of you know, this is what we do in patients as well, and in human beings and people with mitochondrial disease. and that reduces the reactive oxygen species. And they engineered the mice to express an ROS natural scavenger called catalase, and that reduces the reactive oxygen species. And what they found here was that increased reactive oxygen species was associated with a lower diversity. So lower diversity of the gut microbiome, dysbiosis of the gut microbiome. And when you improved the reactive oxygen species, that's to say you lessened it, the gut microbiome diversity improved. So this is in mice. And then a couple years after that, my boss, Dr. Eva Morava, who's chief of metabolism here at Mount Sinai, and Dr. Kozics decided to look at this in some adults with MELAS. So only a handful of patients, about 7 or so, but you can clearly see here that compared to closely matched controls, so that's age and household matched controls, there is a clear difference between patients with mitochondrial disease, in this case MELAS specifically, and they looked at the alpha diversity here and the Shannon index here. So you're seeing two things here. Not only is there an obvious apparent difference between the gut microbiota and the diversity in controls and in patients with MELAS, but we see consistently across both Shannon and alpha diversity that patients with mitochondrial disease overall have lower diversity of their gut microbiome. So this is clinically significant because, as we talked about, dysbiosis of your gut microbiome can be associated with so many clinical consequences. Anything from malabsorption, malnutrition, different neurotransmitters in the brain, disease states like diabetes. The gut microbiome and the immune system are very tightly regulated, are very tightly associated. And so dysbiosis in the gut microbiome may also lead to long-term chronic inflammation and neuroinflammation. And as many of you know, that's also in its own right an emerging area of research in mitochondrial disease, where we're looking at how the innate immune system, the interferon signature pathway in our body, it is tightly associated with different states of different genotypes and different phenotypes of mitochondrial disease. So what else affects the gut microbiome? So obviously, I hope you've seen from these last several slides now that the mitochondria and the mitochondrial DNA and the reactive oxygen species are all very important when it comes to looking at the gut microbiome. But there are actually many factors that can alter our gut microbiome. So one thing is diet and nutrition, so macronutrient balance. So we already know that this is increasingly important in primary mitochondrial disease. Recent studies from CHOP, for example, have shown that about a quarter of patients with mitochondrial disease in adult cohorts meet criteria for malnutrition. And as many of you know, a lot of children with mitochondrial disease experience lack of appropriate weight gain and difficulty gaining weight, and many GI issues as well. And what they found also, what we're finding is that in patients who are not meeting their macronutrient goals, and that means not meeting their recommended daily allowance for carbs, fat, and protein, improving those goals, making sure patients reach those goals in macronutrients like protein and fat, were thought to lead to and be associated with improvements in fatigue, improvements in strength, and improvements in quality of life as well. So we know, and this is not surprising, right? We know that the mitochondria generate energy from the food we eat. And so it's very reasonable to think that nutrition plays a very important role in mitochondrial disease, and now perhaps increasingly so when we also take the gut microbiome into consideration. Medications, so many patients with mitochondrial disease are on multiple medications, and many medications beyond just antibiotics can alter the gut microbiome as well. Age, so older patients and younger patients have different sets of gut microbiota as well. We talked about the immune system and gut microbiome being associated, and this is something I'm particularly interested in. So the questions I asked myself after this were, can the gut microbiota in patients with mitochondrial disease tell us anything about clinical prognosis, response to treatment, phenotype? And then once we answer those questions, what are targeted treatment implications? What are the effects, if any, of manipulating diet and nutrition to improve gut microbiota? What can we do in terms of prebiotics and other factors that we could employ to manipulate gut microbiota. How can we target reactive oxygen species to improve gut microbiota, and so on and so forth. And I was particularly interested in looking at this in children and with Leigh syndrome. So I'm very thankful to the Cure Mito Foundation for also being very encouraging and interested in this study and funding a pilot study for this. So what we're looking at, and this is just a pilot study to start, we're looking at this in patients with ECHS1, SIRF1, and MTATP6. And those are not randomly chosen. We chose these because the three of them together give us different degrees of reactive oxygen species. So MTATP6, for example, might be associated with higher reactive oxygen species. ECHS1, through buildup of toxic metabolites, suppresses the respiratory chain and also can increase ROS to a sort of middle ground. And then SIRF1 is associated with less ROS. So we're going to look at the gut microbiome in each of these patients' populations. And this way we also have nuclear contributors and MTATP6, a mitochondrial genotype as well. So we're looking at both nuclear and mitochondrial DNA. For each patient we recruit and look at and examine, we'll look at the gut microbiome of an appropriately matched control. So age matched, household matched, and so on. So if you're interested in participating in this study, what it'll involve is setting up a remote meeting with me to go over the consent form, over the information, and obtain virtual consent, sending medical records, so genetic information, medical notes, what medications the patient's on, and so on. If possible, completing a 3-day dietary log, because we want to look at how nutrition might affect the gut microbiome, or what we're seeing when we investigate or interrogate the gut microbiome. And then lastly, just mailing a stool sample in a kit that we mail you with a stool sample collection kit, mail back to us here at the Icahn School of Medicine, where we will sequence it and analyze it. So if interested, please email Casey or Sophia or myself, and we'll be in touch as soon as we are ready to begin active recruitment. We just got IRB approval to begin, so we should be ready to launch in a few short weeks. So thank you. Very grateful for the Cure Mito Foundation and my mentors here at the Icahn School of Medicine at Mount Sinai, and for you all as well for participating in research like this. Thank you.
Thank you so much, Dr. Elsharkawi. That talk was very clear, and I think that the gut health really resonates with all of us. There are so many factors that influence it, and it's exciting that you're addressing that from a very practical perspective in a pilot study. So thanks for that.